We know that the genetic code is a series of nucleotides that is brought to the ribosome by the mRNA. Here, it is converted to protein with the help of tRNA. We also know that each protein has a unique sequence of amino acids which make up its primary structure. This is important because the sequence of amino acids dictates how a protein folds and subsequently its functionality. Nuremberg and Mathieu determined that certain RNA sequences code for certain specific amino acids. To do this, they set up 20 test tubes, each with a cell-free environment, and a different amino acid in each one. The amino acids added to one test tube were radioactive, so that any polypeptide formed would be more easily noticeable, and that amino acid was phenylalanine. They synthesized mRNA that was comprised exclusively of uracil, called poly-U. This was then added to each of the test tubes. The test tubes were then incubated so that translation could occur. Afterwards, in the test tube with the radioactively tagged amino acids, they discovered a newly formed protein that was exclusively comprised of the amino acid phenylalanine. This meant that the codon UUU coded for phenylalanine, and also told us that there was a code which could be cracked. Eventually, these tests were repeated with poly-A, poly-C, and poly-G. This led to the discovery that AAA coded for lysine, CCC coded for proline, and GGG did not code for anything. In another experiment, Nuremberg worked with Lederer to determine the rest of the genetic code and figured out which codons coded for which amino acids. They once again set up a test tube with a cell-free environment that just contained ribosomes and charged tRNA. This is tRNA with amino acids attached. They made one amino acid radioactive. They then synthesized mRNA that was just one codon long and added it to the mixture. The ribosome binds the mRNA to its corresponding tRNA along with its attached amino acid. They then filtered out the unbound tRNA and tested the filter paper for radioactivity. If the filter paper was radioactive, then the mRNA codon they had added to the test tube coded for the radioactive amino acid. They did this procedure to identify 61 separate codons.